Hello everyone, welcome to the series of lectures on reproductive biology. And before understanding the concepts of reproductive biology, we must first understand about the reproductive endocrinology. And here I will bring few slides wherein we will understand and see the contribution of scientists who have contributed remarkably to this field. So in 1849, Arnold Adel Berthold, he explained at the Royal Scientific Society an experiment which he conducted using only 6 cocks. He took 4 cocks and castrated them, that is removed their testes, whereas he kept 2 cocks as control wherein he did not perform any surgery of removing the testes. So as expected, it was seen that these 2 uncastrated cocks, they developed normally, whereas if we see these 4 cocks which were castrated, there were no male characteristics. Now what Berthold did, he then further divided these 4 cocks into 2 groups. In 2 of the cocks, he transplanted the testes back into the abdomen, wherein he observed that these cocks developed normally and had normal male secondary sexual characteristics, whereas the remaining 2 cocks wherein no testicular transplantation was done, you can see in the picture that there were no male secondary sexual characters, there was no crown at the top and the body of the male was also bent. So this suggested that probably this testes is contributing to some factor which is responsible for the male sexual characteristics. Thereafter in 1927 and 28, Wieland and Windaus, they both got the Nobel Prize for discovering the chemical structure of bile acids, cholesterol, sterols related to vitamins respectively. Soon, an experiment was conducted by Brown-Sequard wherein he showed that adrenalectomy if performed would be fatal to the lab animals. He also performed another experiment wherein he kept himself as a subject. He self-injected the elixir of juice extracted from the testiculars of dog and guinea pig, and he said that it is the elixir of life and it gave a lot of energy. Thereafter, Thomas Edison, who is also referred to as the father of endocrinology, he actually determined the Edison's disease, the cause behind it, and showed that adrenal destruction is fatal in humans. Soon after, a number of labs started working on finding out these hormones or the factors which are produced by the adrenal. And it was isolated that there are so many steroid hormones produced from the adrenal gland wherein there are only minor differences at hydroxyl and ketone groups at different locations. And out of this effort only, the life-saving drug came into the picture, and that was cortisone. Soon after, in 1932, Rosenheim and King, they determined the accurate structure of steroid nucleus, wherein there are 4 ringed structures labeled as A, B, C and D. This is the steroid nucleus, also referred to as cyclopentanoperhydrophenanthrene nucleus, which remains common for all the steroid hormones which we are going to understand today. So in 1939, Nobel Prize was shared by Leopold Ruzicka and Adolphe Butenandt for determining the correct structures of estrone, progesterone and androsterone. Now importantly, all these hormones are produced from a 27 carbon containing precursor, that is cholesterol. So here you have to note the position of the carbons 1, 2, 3, how this A ring contains 5 carbons, the B ring contains another 5 carbons, then 11 to 14 are there in C carbon chain, and then in the D ring there are remaining carbons. Then after the 17th carbon, here you can see that 18th and 19th carbons are actually the methyl groups. And then there is this ring, this chain of the hydrocarbon, which is making it a 27 carbon compound called as cholesterol with OH group at the third position and double bond between fifth and sixth carbon. Now the question arises that from where do we get all these cholesterol? Do we obtain it through diet only, which contributes to steroid hormones? So actually you are correct that you definitely derive it from the diet, but in our cells there is de novo synthesis of cholesterol as well, which is shown in this pathway. So from acetyl-CoA there is the conversion of it into hydroxymethylgluteryl-CoA, which by the action of HMG-CoA reductase gets converted into mevalonic acid that in turn enters into the mevalonate pathway. Now after that it gets converted into geranyl pyrophosphate, which then chooses two parts. One is for the prenylation of protein, the formation of isoprenoids occurs, and in the other pathway by the action of several synthase and oxidase enzymes, there is the formation of cholesterol by the intermediate formation of squalene, lanosterol, and a number of reactions take place which gives rise to cholesterol. Now once cholesterol is synthesized, which takes place in the endoplasmic reticulum, now subsequent steroid hormone synthesis has to be done in the mitochondria. So now there is another question that how would this cholesterol cross the membrane of mitochondria? And here comes an important protein named as star protein. This star protein, which stands for steroidogenic acute regulatory protein, it helps in the transport of cholesterol from outer mitochondrial membrane to the inner mitochondrial membrane. So now the cholesterol basically moves towards the inner mitochondrial membrane, wherein it meets with another set of enzymes, which leads to the conversion of cholesterol to pregnenolone. Now here an enzyme called P450 side chain cleavage enzyme acts, which cleaves this entire chain, resulting in the formation of 21 carbon compound called as pregnenolone, and a bicarbonate is formed called as isocaproaldehyde. Now interestingly, this reaction takes place in the mitochondria. It is not a one-step reaction, rather in step 1 hydroxylation occurs at 22nd position, resulting in the formation of 22 hydroxycholesterol, followed by another hydroxylation at 20th position, giving rise to 2022 dihydroxycholesterol, and finally there is the cleavage of this chain giving rise to the formation of pregnenolone. Now this reaction is important for one more reason, and that is that it is a rate limiting step which determines the synthesis of rest of the hormones, and it is under luteinizing hormone regulation. And this step takes place, as I have told you, in the towards the inner membrane of the mitochondria. Now once pregnenolone is formed, there are two fates of pregnenolone. Now what are those? In the first fate, pregnenolone is converted into progesterone and is called as delta 4 pathway. Now here I would request you to please note the position of the double bonds. Now here what will happen, there you can see in the pregnenolone the double bond is between 5th and 6th carbon. But by the action of an enzyme called as isomerase, there is transfer of this double bonds between the 4th and 5th carbon. And then there is one more reaction which is taking place by the action of dehydrogenase enzyme, that is 3-beta hydroxysteroid dehydrogenase, 3-beta HSD. You can see at the third position there is OH in the pregnenolone, but here by the action of dehydrogenase enzyme you can see that there is ketone group now. Rest of the structure remains the same. Hence pregnenolone gets converted into progesterone in delta 4 pathway, since the double bond has shifted towards the 4th and 5th carbon, and this reaction, please note, takes place in the smooth endoplasmic reticulum, and is predominant in corpus luteum and placenta, wherein progesterone synthesis takes place. In the second fate, which is called as delta 5 pathway, because you will see that in all these three compounds, the double bond, positioning of the double bond does not change and remains between 5th and 6th carbon. And here there is formation of from the pregnenolone, it will get converted into 17 alpha hydroxy pregnenolone by the action of 17 alpha hydroxysteroid dehydrogenase. So, interesting thing here is that this 17 alpha hydroxysteroid dehydrogenase actually has two types of activities. First is hydroxylation at the 17th position. So, please see, I have marked the 17th carbon. Here you can see everything is same, then all the structure, entire structure is same, but at the 17th position there is addition of OH group. So, this is hydroxylase activity of 17 alpha hydroxysteroid dehydrogenase, resulting in the formation of 17 alpha hydroxy pregnenolone. Now, by the action of same enzyme, but it is another activity which is called as C1720 lyase activity. Lyase means it will break now the bond between 17th and 20 carbon. So, resulting compound would be DHEA, which is dehydroepiandrosterone. So, here in this pathway there is involvement of 17 alpha hydroxysteroid dehydrogenase, and here it requires the cofactors, namely NADPH and oxygen. Now, DHEA, please note, is the first androgen which I am showing here. And please note that now the carbon number has further reduced to 19 only. So, progesterone, pregnenolone, all they had 21 carbon, but the androgen has only 19 carbons in it. And this reaction is predominant in adrenal cortex part. Specifically, if you could recall the endocrinology series, we have talked about different parts of the cortex. There, the innermost part of the cortex is the zona reticularis, which gives rise to the DHEA and definitely the gonads. Now, what happens next? So, we have seen that pregnenolone gives rise to DHEA by the action of the enzyme called as 17 alpha HSD, and we have also seen that pregnenolone can give rise to progesterone by the action of 3 beta HSD and isomerase enzyme. Now, this progesterone gets converted into 17 alpha hydroxyprogesterone by the action of same enzyme which we have studied here, that is 17 alpha HSD. So, you can see the structure is entirely same, but OH group gets added at the 17th position. Now, the same enzyme will act, but if you could recall, by the action of lyase activity, there will be formation of the another androgen, which is androstenedione. Now, there you can see in this structure there are two ketone groups, and that is why it is referred to as dione. Two ons are there. So, now we have with us DHEA as well as androstenedione. I would like to take your attention towards this step also, that 17 alpha hydroxy pregnenolone and DHEA, both of them can get converted respectively into 17 alpha hydroxy progesterone as well as androstenedione by the action of 3 beta HSD and isomerase enzyme, which we have studied here in the pregnenolone to progesterone conversion. So, now you have androstenedione with you. By the action of another enzyme called as P450 aromatase, this androstenedione gets converted into the first estrogen, which is E1, estrone. Now, why it is called as E1? Because there is only one OH group, and by the action of aromatase enzyme, there is aromatization in the A ring. You can see that. Now, this A ring gets aromatized. At the third position, there is OH group added, and one more change you have to take attention of or take note of it is that there is loss of one methyl group also. Now, there are only 18 carbons in estrone. Now, if you look further, once estrone is formed by the action of 17 beta HSD, yet another HSD, 17 beta HSD, this estrone can get converted into estradiol. So, dehydrogenase activity, you can see that here the O group, the ketone group gets converted into the hydroxyl group. Now, there are 2 OH and hence estradiol is formed. Now, once estradiol is formed from estrone, you have to note here that this androstenedione can also get converted into testosterone by the action of 17 beta HSD. And from your school time, I am sure you have studied that estradiol is derived from testosterone. So, you have seen in this pathway that how these androgens are getting formed. Androstenedione is forming. Now, this androstenedione can get converted into testosterone, and testosterone then gives rise to estradiol by the action of the same enzyme which you have studied, that is P450 aromatase. Now, once estradiol is formed, this is the primary hormone which regulates the female secondary sexual characters. Now, once testosterone is formed, it can get converted into 5 times more potent androgen, which is called as dihydrotestosterone by the action of 5-alpha reductase enzyme. So, you can see that because of the 5-alpha reductase enzyme, there is loss of double bond between the carbon of the ring. Now, you can see that there are 19 carbons here. OH group is there as there was in testosterone. Rest of the structure is same, but there is loss of this double bond. This is giving rise to the 5 times more potent androgen, which is called as dihydro. Please note, it's not dihydro, it's dihydrotestosterone. So, this is the entire pathway of steroidogenesis, which I have brought here on one page, from wherein you can see that how cholesterol is getting converted into pregnenolone, and pregnenolone is thereafter entering into 2 different fates. At one point of time, it can give rise to progesterone, whereas on the other hand, it can form DHEA, that is dehydroepiandrosterone. Then this progesterone can also follow the same path as pregnenolone followed, and how this progesterone is giving rise to androstenedione. Then androstenedione further can enter into 2 pathways. One is the formation of estrone, and then it can also give rise to testosterone. So, from testosterone and estrone, we have seen that how can estradiol be formed, and this testosterone can further get converted into DHT by the action of 5-alpha reductase. So, this is a summary picture wherein you can see that all the compounds getting formed. So, at the end of this lecture, which dealt with the historical perspective of the discovery of those hormones which play a significant role in reproductive biology. Apart from the historical background, I have explained the steroidogenesis pathway, wherein we have seen that cholesterol, which is a 27 carbon compound, is getting formed within our body, which is called as de novo synthesis of cholesterol, and then there is another way of by consuming through diet, cholesterol can be obtained. This cholesterol, once getting formed, there is another important role of a protein called as star. There are several studies wherein if star gets mutated, there are number of deficiencies related to these sex steroid hormones, and I am sure now you can relate why it is so. So, this star, which stands for steroidogenic acute regulatory protein, it plays a significant role of transporting cholesterol from the outer mitochondrial membrane to the inner mitochondrial membrane. And then what you have to keep in mind is that progesterone, pregnenolone, they all belong to a family called as progestogens. They all are 21 carbon compounds. Whereas if you talk about the androgens, they are 19 carbon compound, and then estrogens they do contain only 18 carbons. And if you talk about the site of production of these sex steroid hormones, you have seen that there is implication of both mitochondria as well as smooth endoplasmic reticulum. So, these are different sites where the steroid hormones get produced. So, in this initial series of the reproductive endocrinology lecture, you have understood about these synthesis of the hormones, and in the next lecture you will understand how these hormones perform their action. So, the mechanism of action of these hormones would be shortly explained to you in the next lecture. Thank you.